Right. Hi everyone, welcome to lecture 8 of BIS103. This video will be on the Entner-Duderov pathway. So our learning goal for this video will be to explain at least the principal reactions of the Entner-Duderov pathway and how we can compare it to glycolysis. The Entner-Duderov pathway is actually an evolutionarily ancient pathway. We find it in evolutionary older organisms such as archaea, gram-negative bacteria, and a few select gram-positive bacteria. And what it actually is that it's a combination of our glycolytic pathway and the PPP. How does this work? If you actually compare it to the two pathways that we covered in this lecture, the PPP, and in previous lectures on our glycolytic pathway, if you look at this first reaction here, glucose to glucose 6-phosphate, that very much looks like glycolysis, right? That's our first reaction also of glycolysis. And at the very end here of the Entner-Duderov pathway, we have our reaction from G3P to pyruvate, right? That was our payoff phase of glycolysis. So we have a few parts of glycolysis in this pathway. And then if you look in the middle here, which are highlighted here in red, again, the beginning, glucose to glucose 6-phosphate, and then specifically from glucose 6-phosphate, the oxidation to the lactone here, to the phosphogluconodelta lactone, and the next reaction here of the hydrolysis to the gluconate, so the carboxy acid of glucose 6-phosphate. These were our entering reactions of the PPP, right? So we have a combination of these two pathways here. And then in the Entner-Duderov pathways, in these organisms have just two additional enzymes that are key here to combine these two parts of the PPP in glycolysis. And they are highlighted right here in purple. What we're actually doing is that first we're doing a dehydration at this position here, right? We're removing the hydroxy group to this molecule here. You don't have to remember the names of metabolites or the structures at all. Just look at the general reactions. And then the next step is a cleavage, a carbon-carbon cleavage reaction, much like we had seen in our priming phase of glycolysis. But with a major difference here is we're not generating DHAP and G3P that we then can convert into two molecules of G3P by isomerization. We're actually generating one molecule of G3P and we're making one molecule of pyruvate. This has really important consequences for our ATP yield, right? So the net ATP yield of the Entner-Duderov pathway actually is only 1. And that is because, right, we are making in this part of the pathway here, right, G3P to pyruvate. Right? We had talked about our substrate level phosphorylation, we're making 2 ATP. We're using 1 ATP in this first reaction here. So 1 ATP. And what we're not doing, like in glycolysis, is that we double up on our payoff phase, right? Our second product of the carbon-carbon cleavage reaction is not G3P, it's pyruvate. So we jumped already all the end to glycolysis here. We are bypassing the possibility to do any further ATP production. So key take-home messages, it's a very ancient pathway. It's a combination of glycolysis and the pentose phosphate pathway with 2 additional enzymes. It has a lower ATP yield, but it allows you to utilize a combination of these pathways to do and to prepare ATP. What I want you to know about this pathway really is only these general reactions as we discussed them in this video. I don't expect you to learn the structures, nor do I expect you to learn the enzyme names. If you can understand these different general reactions, how they compare to glycolysis in the PPP, and how that impacts ATP yield, that will be perfectly sufficient.